Hello, everyone. Welcome back from our short break. I hope that you're enjoying the symposium so far. My name is Thomas Wilmansky, and I'm a postdoctoral research fellow here at ISB, and I'll be moderating the last session of the day, session 3 in VIVO interventions. We have two really fascinating speakers, Dr. Gautam Dantas and Dr. Joanna Lampe. As a reminder, please ask your questions via Zoom through the Q&A chat room. Each presentation will once again last 30 minutes, and it'll be followed by a joint 30-minute Q&A session afterwards. Our first speaker of the session, Dr. Dantas, is a professor in the Department of Pathology and Immunology, the Department of Biomedical Engineering, the Department of Molecular Microbiology, and the Edison Family Center for Genome Sciences and Systems Biology at the Washington University in St. Louis School of Medicine. He received his PhD in biochemistry from the University of Washington under the guidance of Dr. David Baker, and his postdoctoral training in microbial genomics from Harvard Medical School under the guidance of Dr. George Church. Dr. Dantas' research interests and training lie at the interface of microbial genomics, synthetic biology, systems biology, and computational biology. His current research focuses on understanding and predicting how diverse microbiomes respond to chemical and biological perturbations, harnessing these insights to rationally design therapeutic strategies to curtail antibiotic-resistant pathogens and remedy pathological microbiome states, and engineering microbial catalysts to convert renewable biomass into value chemicals such as biofuels and pharmaceuticals. He is the recipient of several different awards, including the American Association for Advancement of Science Newcomb Cleveland Prize, the Harvard University Certificate for Distinction in Teaching, the NIH Director's New Innovator Award, and the Kenneth Rainin Foundation Breakthrough Award. Today's talk by Dr. Dantas is titled Predicting and Combating Pathogenic and Abiotic Disruptions to Diverse Microbiomes. Please join me in welcoming our first speaker, Dr. Dantas.
Great, thank you, Tomas, and thank you for the rest of the folks at the ISB for giving me this opportunity to tell you about some of the work in our lab looking at how sort of bugs and drugs interact with microbiomes. But before I get going, I think it's really important for me to acknowledge that I'm merely a spokesperson here. Anything of interest or importance that I might talk about today is thanks to the incredible group of people that I get to work with and represent. While I will highlight a couple of people who are responsible for a couple of the key vignettes, also please realize that all of those are team exercises, both with others in the lab as well as our many excellent collaborators. So the topic of my presentation today is this concept that bugs and drugs both alter the microbiome, but understanding those effects and predicting them requires an appreciation of context. And so we recognize that both drugs as well as bugs, so things like antibiotics and pathogens, can be destructive to the microbiome. But then there are also restorative potentials for so-called microbiome-directed medicines, for things like fecal microbiota transplants, but also engineered or chosen probiotics. And it's really the question of how do we take into account the diversity and the composition, as well as features like diet, which I presume we'll hear in our second talk, and a variety of other environmental conditions that impact how those destructive or restorative properties manifest. And there's rich literature to show that both in this case, animal models show that dysbiotic guts in terms of low diversity microbiomes can enrich for pathogen infection. And then also in human studies, where here in a pair of papers, these authors showed that different microbiome compositions of individuals drives differences in probiotic colonization resistance. So let's start with antibiotics as one of these big features that can perturb the microbiome. We think of antibiotics as small molecules that target pathogens, but the reason antibiotics work is because they target all of these key cellular processes of bacterial life, which, as it turns out, are shared by both good and bad bugs. So effectively, as you ingest an antibiotic, you're hitting as many good bugs, if not more, as the infection itself. And indeed, through retrospective analysis, essentially at any part of the age spectrum in human life, there are a whole series of comorbidities associated with antibiotic use. The greatest comorbidity, of course, with antibiotic use is antibiotic resistance. And these numbers are probably familiar to all to know that antibiotic resistance encodes an incredible burden, both in terms of lives now as well as in the future, as well as economic costs. And unfortunately, exactly as resistant bugs are on the rise due to antibiotic use, unfortunately, antibiotics are not coming to market. And so with that context, we'd like to understand what heavy antibiotic use does in especially the context of developing microbiomes. So this first vignette is about our focus on the pediatric microbiota, specifically looking at preterm infants, acknowledging and recognizing that as medicine has improved in our ability to keep younger and younger kids alive, it also means that these kids are increasingly more vulnerable to infections and other pathologies because they're born too early. And one of those manifestations is in the microbiome. So we know that age-matched preterm infants versus term infants have a substantially lower diversity of species in their guts, sometimes as much as tenfold lower during those first couple months of life. And layering onto this, when we're thinking of antibiotics, it turns out exactly when this microbiome is getting set up, we bombard them. So we know that, especially very low birth weight infants get antibiotics, virtually all of them during the first couple of days of life, so especially if they're born about 10 weeks too early. And over the course of your stay in the neonatal ICU, where these kids are hospitalized, the top 2 drugs and 4 of the top 8 drugs that they receive are antibiotics. So in collaboration with Phil Tarr and Barb Warner at Washington University and St. Louis Children's Hospital, we've been studying for the last 10 years or so the impact of antibiotics in the developing microbiome. So in this first study, Molly Gibson, a graduate from our computational biology program, teamed up with Barb and Phil to look at a cohort of preterm infants who are hospitalized for their first 2.5 months or so of life. We had fecal samples available, and Molly stratified this cohort into a group of kids that only had antibiotics early in life versus a set of kids who had antibiotics early and then often. And through a combination of metagenomic interrogation techniques, she discovered that the bugs that dominate the guts of these kids due to antibiotic selection pressure tend to be the type of microbes that we don't really associate with good gut health, but rather often pathobionts, or at least the type of bugs that we might think of as causing opportunistic infections. But through a combination of microbiome and antibiotic resistance profiling and application of machine learning approaches, Molly was able to show that by training random forest models on microbiome samples available before treatment, she could predict the outcome of treatment in terms of microbiome diversity based on only a few variables, in this case, 2 antibiotic resistance genes and 2 bugs. And so what this says is that even though there are these acute damaging features of antibiotics in the microbiome, maybe there's a tantalizing future possibility that we can use microbiome-based diagnostics to understand how the context, that is the microbiome, might tell us how antibiotics might impact it. So with that acute information in hand, we wanted to ask, what is the potential long-term impact on the microbiome of these kids, especially after they've left the neonatal ICU and they've got other environmental factors come into play. So to answer these persistence questions, again, teaming up with Barb and Phil, but now with work led by Drew Gasparini, a graduate from our lab in the genomics program here at WashU, we had access to a series of outpatient samples from these same kids out to about a couple of years of life. And so Drew applied a series of similar microbiome interrogation techniques, now having 3 cohorts to investigate. So what you're seeing here is the number of treatments that these kids get cumulatively. So obviously you've got this heavy antibiotics perturbed group of preterm infants. You've got the infants who get antibiotics only early in life. And then we added on a 3rd cohort here of term infants born in the same hospital who had no antibiotics at all, sort of our healthy, normal controls. And so through a combination of shotgun sequence to interrogate who's there in the guts of these kids, functional metagenomics, this method allows us to identify both known and unknown antibiotic resistance genes, and some deep culturing and genome sequencing of particular drug-resistant isolates, we were able to begin to answer this question of potential persistent effects. In work that I don't have time to go through, I should also mention that while these human observational studies are very powerful for generating hypotheses, they're unable to actually test hypotheses explicitly. And so one of the things that we're doing now is we're using a series of transgenerational transfer humanized microbiome germ-free animal experiments where we introduce those same fecal samples from this human cohort and then kind of run in replicate the same type of features we see in the human cohort to try to improve our predictions. And that's work that's being led by Drew Schwartz in the lab, follow-up on work that Terence Croft and ex-postdoc started. Okay, so what Drew found in this particular study of infants similar to Molly's study, except now with outpatient samples, was some interesting trends in terms of comparing the term kids, who, and this is the resolution of months. The diversity of these kids, as you might imagine, starts very low and then jumps up pretty quickly in the term kids, and immediately over the first couple of months sort of achieves the type of diversity you'll see towards the end of the first year of life. Whereas the trajectory for the preterm kids is much more shallow. By 12 months of age, they seem to be at about the same diversity, but it takes them a long period of time. So this sort of developmental delay, if you will, in their microbiome. Of course, the devil is in the details, and I won't belabor each of these stacked bar histogram points, except to say that diversity is not the only measure of importance in terms of microbial composition, such that, you know, we might see, even if the diversity looks like it's similar, you might see features of this type. For instance, here, the preterm cohorts, these green bars correspond to Enterococcaceae, and these MDR versions of these particular bugs are completely absent in these term infants, which you might miss if you were only focusing on diversity. And similarly, on the other side, you look at this particular light green set of bars in the term infants, not really present here in the preterm infants, responding to these particular Prevotella. So also showing you that while you might have diversity catch-up, you may not have compositional catch-up. Now focusing on resistance genes, this other sequelae of antibiotic use, Drew applied this particular technique of functional metagenomics, discovered many thousands of unique resistance genes in the guts of these kids. And interestingly, the point I want to make here is that because he used this method that is initially agnostic to knowledge of resistance genes, it emphasized that even the best resistance databases out there, for instance, this comprehensive antibiotic resistance database, only a third of our genes could really be found by comparison to that database, and whereas all of the genes we found had close homologs, high identity hits in NCBI. So what this tells us is that this method of functional metagenomics, is capturing genes that other people have sequenced before, but they haven't identified as resistance genes. So it's telling us that this very clinically important feature is still massively underrepresented in our, at least, database representation of resistance. And perhaps unsurprisingly, when we look at what type of bugs have these particular resistance genes predicted, the top two bugs are uncultured bacteria and bacteria unclassified, obviously not terribly informative there. So emphasizing we don't know the bugs, we don't know their functions. And in the cases where you do know what the bugs are, they're, again, those type of opportunistic pathogens, not the type of things we want in the guts of these kids. Then we wanted to ask, okay, we know that the resistance is there. How does it seem to segregate across these cohorts of kids who are variably antibiotic perturbed? Interestingly, when we look at unweighted metrics, so that is looking only at the presence or absence of resistance genes and their unique numbers, we find that they're fairly similar across the three cohorts. That is to say, in fact, we might even see a few more unique resistance genes in the guts of these term infants who had no antibiotics. But perhaps now intuitively, you might expect that while you're not changing the sort of playing field of which type of resistance genes you have, what you're really changing is selection pressure for the enrichment. So this is to now say, if you look at the abundance of the resistance genes, they're way higher in the guts of these kids who had lots of antibiotics early in life. So, maybe not a groundbreaking discovery, but something that really affirms the idea that antibiotic use is not inventing new resistance genes. It is enriching from existing reservoirs, and it does so in a manner that is consistent with antibiotic use. Then the other thing that Drew wanted to do in terms of validation was to focus in on these particular bugs that Molly had originally identified as being prevalent and abundant in the guts of these kids. He also found the same thing. This is early in life. And what he wanted to ask was that perhaps early enrichment during antibiotics going to cause these bugs to kind of get stuck in a niche and then still be persistently colonizing them? So what he did was he looked at fecal samples, time matched, or I said age matched across kids, samples taken during hospitalization, and then about 8 to 10 months later, and then trying to culture for each of these type of bugs. And what he found was, in a number of cases, by isolating bugs and then sequencing their genomes, found a number of these bugs that appeared to be identical both at these early and late time points in the antibiotic perturbed kids, but not so much in the healthy infants. So again, now providing evidence that in addition to antibiotic resistance enrichment, you're beginning to see these potential scars, if you will, of early antibiotic use, the persistent colonization enrichment of drug-resistant bugs. Just a quick aside to make sure that we're not over-interpreting this data. This data doesn't say anything about pathogen enrichment. These are not necessarily pathogens. These are just drug-resistant commensals that could potentially cause opportunistic infections given later disturbance in the gut microbiome. Okay, and then finally, to be able to ask whether these features could be detected at the sort of microbiome molecular feature level, what Drew did was he trained a support vector machine, fancy word for a pattern recognition algorithm, on the 16-month samples for the term versus the preterm kids to ask if you were to train a model on these two sets of microbiomes using both taxonomic as well as resistome data, could you distinguish whether those kids were born preterm or with antibiotics? Now, I will somewhat facetiously say that that is a very expensive way to figure out if a kid is born preterm, because you could just ask their mom or dad or a primary caregiver. That's not the deliverable. The deliverable is, if you can do this, it again suggests that there are molecular features, scars, if you will, that distinguish these microbiomes. And sure enough, that's what Drew was able to show. With some feature selection and enrichment, he was able to identify 15 features, roughly half and half resistance genes and particular taxa, through which he could, with 96% accuracy, classify these particular microbiomes. Again, suggesting that there are these features that tell you whether a kid has gone through those early perturbations and perhaps setting you up for interventions in the future. So with that, it's a kind of nice transition towards what can we do if we wanted to perhaps remediate some of these issues. And one such strategy is probiotics. So we've gone in a sense, with what we've shown just before this, and kind of wrecked the microbiome by using the language of warfare rather than the language of microbes in remediating states. So what if you instead use the language of microbes, right? And again, I would argue that perhaps the best examples of this are the very successful use of fecal microbiota transplants for something like Clostridium difficile. But what if we wanted to get a little bit more defined, a little bit more specific with probiotics? So there are a number of recent forays into more defined and engineerable probiotic organisms. And one such organism is a strain of E. coli called E. coli missile, originally discovered by a German physician in the First World War as a bug that he found from a soldier that happened to survive a diarrheal attack of the rest of the troops. And so there are a variety of recent academic as well as commercial ventures into taking our sort of favorite model organism E. coli and using all of the genetic tools available to train them on this missile strain to have it sort of be a drug delivery vehicle for interesting therapeutics. But there are a couple of challenges along the way in terms of considering how do we treat this bug, which is ultimately a living, breathing, or perhaps breathing, evolvable organism compared to how you might treat a small molecule chemical. So one of the issues to consider is that, again, unlike aspirin or paracetamol, you might end up getting colonization of this particular organism, and you might have it happen variably. So this is data from a clinical trial that shows that after initial treatment in humans, up to 24 weeks later, there was still a couple individuals who were colonized with this bug, but it was highly variable across the cohort. And then also, and this is not missile work, but in another study similar to the one I showed you earlier, it was shown that particular probiotic cocktail actually made things worse compared to just autologous FMT in terms of recovery from antibiotic perturbation. So this tells us that, yeah, this would be good to have these defined probiotics, but we need to understand how they work. And specifically, we need to account for their living nature as we think of them as engineerable therapeutics. And so ways in which we can do them, we can specifically control them. We can control things like the expression of the therapeutic payload. And we can also make sure that we can put in kill switches or other types of elements which we can use to get rid of the bug after it's done what it's done. But in parallel, by using sort of more basic science approaches, I think we also need to understand how these bugs are potentially changing in response to the selection of the mammalian gut through the processes we know all bacteria used to evolve, namely vertical transmission of sort of Darwinian mistakes in evolution that might occur because these bugs are ultimately reproducing and dividing in the guts of these mammals. But also this very, very important complementary method of evolution of horizontal gene transfer, where the bugs, as they're coursing through the gut, may pick up or donate genes with the human microbiome and change along the way. So to do that, we turned back to this method that we've actually used for antibiotic resistance interrogation called functional metagenomics, and instead decided to use it in this case as a proxy for horizontal gene transfer to understand how a model organism like E. coli Nissl probiotic might pick up genes as it courses through the human microbiome to modify its phenotypes. In this case, we made libraries, so expression libraries from metagenomic samples, some fecal samples of a group of infants and mothers, created libraries that represent about 10 million unique genes. Those libraries were made by extracting this metagenomic DNA, putting it in an expression construct into E. coli Nissl, delivering it to mice, and then seeing basically which transformants survived. And then doing this over time and seeing what type of functions we got at the end. And I'm sort of spoiling the thunder here to say that this was a very successful enrichment, because in most of the selections, we went from these 10 million genes to only about 1 or 10 genes surviving in the end, really saying that this selection was powerful. Okay, so this work was all led by Nathan Crook, who was a postdoc in my lab and now is an assistant professor at NC State, and Aura Ferrero, a graduate student in my lab in the biomedical engineering program. So over the next series of slides, what I'm going to tell you is some interesting findings that we got from all of these series of experiments that Aura and Nathan and their team did, looking at a variety of variables. And so because there are quite a few variables, I'll just define them here. We look at the original library, of course, which I've already defined. We vary things like diet. So you'll see that we sometimes use the mouse diet. Other times we use this so-called Western diet, which is honestly just kind of high-fat, high-sugar cookie dough, which I guess is our Western diet. And we do that both on its own as well as with certain prebiotics. And then another major selection variable is, what is our library competing against? They can just be competing against each other in terms of monocolonized germ-free animals. They can compete against a defined community of human commensals. They can compete against an antibiotic perturbed conventional microbiome in animals, or just the conventional group. And so you'll see these icons over the next few slides, and you might also see this, which is the arms of the study, where usually we have the mouse, we're introducing our library, and then we're looking at fecal samples over time and analyzing them. Okay, so one of the first things we observed, especially in our experiment where it was a mouse chow diet, it was monocolonization, so these are germ-free, so just the library, and to see what happened is we saw that our library very, very quickly bottlenecked the select for functions that E. coli Nissl was using to adapt, that had to do with better utilization of food, right? So essentially a dietary selection. And so what we found is initially through the selection that occurred by sequencing, and so what you're seeing here is effectively particular fragments of DNA that come through and are selected towards the end compared to the million or so that go in. And what we find are these are the contigs that come through from the selection, the bugs that they're likely coming from. And interestingly, in all of these cases, or almost all of these cases, the central gene that is always selected are these, or gene family are these glycosyl hydrolases, which when we subclone them and then we do a series of in vitro tests, show that they're enabling E. coli to use di and trisaccharides, or even bigger, often but have this glucose or galactose, fructose units, and it's enabling those particular transformants to grow better than the wild type. I'll also say that in later experiments, when we also did this with fragments that came from our human chow experiments, and when we even just took this so-called Western diet and just made an extract from it, even that enabled those transformants to grow when regular E. coli Nissl doesn't. So, how do you synthesize all this data? It appears that when the bugs in there, when E. coli Nissl is in the gut and it has no other bugs to compete with, what it does is it likes picking up elements that allow it to be a better utilizer of the food source that's available. And indeed, when we switched to a different diet, in this case, this human diet with or without prebiotics, the story was pretty similar, right? So yes, there were some other unique fragments that came up, but the fragment that always was selected was these glycosyl hydrolases. Not the same glycosyl hydrolase, but from this family that helps you kind of digest sugars. And perhaps this is not all that surprising when we think of the true ecology of E. coli in the context of other commensals, not in germ-free conditions. We know that even though E. coli has a prolific diet, what it normally does is it relies on the action of anaerobic bacteria that tend to do a lot of the digestion of the more complex polysaccharides that tend to give you the simpler sugars that E. coli likes to munch on. And since those were not around, E. coli was going to adapt now to pick up some of those functions. Okay, now let's switch gears a little bit and say, okay, that's kind of what we found with our model for horizontal gene transfer. But we also recognize that E. coli can mutate its own genome, and it might do so, I should say. Constantly, the genome is mutating through Darwinian processes, and if there are appropriate selections, then bugs might change over time. And I think this is a really important thing to keep in mind, because I would say that, in most experiments, including the ones that our lab does, this is something that people basically ignore, right? You look at most microbe studies and even pathogenesis models, no one is really sequencing those microbes at the end of their experiment. They're just assuming what they put in is what they got out, which I would say, at least based on this, is patently false, right? Microbes adapt, and they adapt very quickly. So we wanted to ask what was going on in this particular situation. And fortunately, we had done all of these controls where we did not have our library, we just had E. coli on its own under all of these conditions, and we could ask, over the course of these 5 to 7 weeks, what do these bugs look like? So one of the things that we did was we took about 400 or so of these isolates and sequenced their genomes, and then looked for common mutations that were in mice in different cages and different conditions to identify the type of genes that might be under selection under these particular conditions. I won't go through all of them, but I'll tell you some of the highlights. So one of the things that we find is, again, a parallelism, if you will, in terms of a phenotype that is being selected. And again, that has to do with utilization of food. But now in this case, what we're finding is an example of a gene called NAGC, which is mutated in 10 different ways across almost 20 different isolates and 5 different mice. And what this mutation does is it de-represses the usage of mucin components of this host GLICNAC. And what this really tells you, and again, what you're seeing here are in vitro results that kind of recapitulate this, that apparently what E. coli Nissl is doing is it's mutating aspects of its ability to now utilize a sugar that maybe you don't want it to utilize, right? Which is effectively the inner lining of the gut. Now, again, I'm not claiming like before that this is necessarily turning a bug pathogenic, but it's certainly endowing this bug with the property that we might not think of as useful for a probiotic, or at least maybe useful for the probiotic, but not useful for us. Another mutation, which is in a sense a silly mutation, this mutation is a gene called Kfib, which we know is a gene that Nissl has that is involved in its ability to attach to the host gut epithelium. So as it turns out, because Nissl over many decades of its use in the lab had been passaged in test tubes, had never really needed this gene to be around, so it's going to devolve. It had gained mutations in this gene that knocked out its function. And all we found is that it had gained that function back as soon as you put it in the context of the mouse. So again, the reason I said it's a sort of silly mutation is, the only reason we saw it is because it had lost it under these artificial conditions, but it emphasizes how strong the selection pressure is for properties like attachment to the host epithelium. And what you're seeing here again is just in vitro confirmation using cell lines for attachment with and without mucin to show that that is truly what is going on from that gene. And then finally, and this is one that is sort of close to our hearts, in one of our experiments, we introduced E. coli into conventional mice with streptomycin treatment. And this antibiotic knockdown is a very common thing that's done in pathogenesis experiments, but also in experiments that are done with human trials of probiotics, where they initially do this antibiotic washout. Here again, we give streptomycin only at the very, very beginning, and there's no streptomycin afterwards. And yet, in almost 170 different cases, we found mutations to the gene that gives you low-level streptomycin resistance that persisted over the course of our study. So that is to say, even this one single dose of antibiotic in the very beginning gives you selection pressure for your bug to become more drug resistant, which again is something, especially by the FDA and WHO, is a big feature they worry about with probiotic use in terms of picking up resistance. Okay, so now that we've got an understanding of the trajectory of these bugs, what I'm going to end on the last couple slides is what do we do with this information, right? How do we actually make these engineered probiotics do interesting things? And one aspect of that is to be able to precisely control the expression of genes in their from the genome. Now, we know that a lot of great work has been done in getting promoter libraries engineered in vitro, but how do we know what's actually going to happen in vivo? If we do this one mouse at a time, that's a large number of experiments that we have to do with different types of promoters that would sort of run their expression. And so again now, Nathan and Aura, but in a more recent publication, developed a cool new method. And since it's published, I won't go through all of it, but I'll just tell you that it's a neat method to be able to, in high throughput, measure the activity of a promoter library very accurately so that you can then use it as a tool to control gene expression. And effectively, it involves barcoding your reporter gene, in this case, GFP, and then you can mix all of these different variants where the variant is a promoter library in front of your reporter, and then through sequencing of both DNA and cDNA, be able to figure out what's going on. And so, again, I won't go through all of the details of how this is done, except to show that they came up with a pretty nice method that is predictive of how in vitro measured promoter strengths nicely will predict both stomach and large intestine expression, even though it doesn't do such a great job with small intestine prediction of expression. And then finally, they were able to apply this method to actually engineer and deliver a probiotic into the gut. In this case, a probiotic version of Nissl that is able to degrade phenylalanine, which could be an interesting treatment for this condition, phenylketonuria. And they were able to show that both in vitro and in vivo, this was successful at reducing phenylalanine levels in both male and female mice. So I know we're going to try to stick strictly to time. So just in wrapping up, I hope I've shown you that there are these potentials for understanding how bugs and drugs work better if you take context into account. This is how we look at various times pre-pandemic, but this is probably what more look like during the pandemic. I'll end with, again, acknowledging the incredible people in my lab and those who've gone on to bigger and better things, our wonderful collaborators, both within WashU and without, the folks who generously supported our work. And especially in this pandemic, if there's one thing I can leave you with as a PSA, please use antibiotics only for bacteria.
Thank you, Dr. Dantas, for a very interesting talk. Dr.